So I'd like to introduce all of you to my Uncle Billy. And as you might be able to tell, he loves to ref youth basketball all the time. But unfortunately, he's had to slow down a little bit because he was diagnosed with cancer. And this is a story I'm sure all of you are way too familiar with. So there was an option for him to join a clinical trial, and everything seemed great. He joined the clinical trial, and the drug worked really well for him, but only for a little bit. And then back to the hospital he went, and it was another clinical trial and another, and they all worked really well for a little bit. And so now he's still on another clinical trial, and we all hope, this will be the one that works, and he won't have to try again. But unfortunately, that's not always the case. And so as a tissue engineer, I can't help but wonder, why aren't we developing treatments for Uncle Billy based on his cancer instead of just trying out all these clinical trials? Like, let's develop his treatment based on his cancer. And so now you might think, I doubt Pharma is going to develop a drug just for me or just for you or just for my Uncle Billy. But it makes a lot of sense when you think about the whole drug development process as a whole. It takes years and years to get a drug to market and billions of dollars. And if out of the 9 drugs that finally make it to the clinical trial stage, 8 of them ultimately fail. And they're failing for one very simple reason, and that's just that we're not rodents, we're not animals. And so all these drugs that are done, spending tens of years and billions of dollars on animals, fail to predict how the drug will work in the intended patients as humans. So by being able to predict whether this drug will work in humans and work effectively, you can decrease all these costs. And so then it becomes more of a realistic scenario where we can actually develop drugs, for us individually. And so what's the solution? What's better than animal models? And luckily, we can use human models. And so with patient-derived human-induced pluripotent stem cells, we have that capability. So what are these induced pluripotent stem cells? So these are cells you can get from your hair, your skin, your fat, your blood, and then you induce pluripotency or trick them into becoming a stem cell. And then the stem cell can become any tissue within your body. And so if you don't believe me, I'll show you. This is Holly. So Holly works in the lab with me, and she donated a little bit of her blood, and then we took this blood and we made Holly's induced pluripotent stem cells. And from her stem cells, we made her cardiac cells, and they're right there beating in the dish. But it's not, just cells beating in a dish we want, because just like animal models don't work, neither do these single cell cultures beating in a dish. So we need to organize them into more complex structures so they're actually acting like a tissue. We want to make the minimal functional unit for each organ. So for every organ there is, what can we recapitulate in the lab that mimics how it functions in the tissue? And I'm going to keep talking about cardiac tissue engineering because that's my specialty, but you can do this for any tissue and every organ in your body using the same similar principles. So to make these human hearts in a dish that actually act like the hearts in our chest, we need to engineer the cell environments. Cells already come pre-coded with this blueprint. They've got their genetic DNA. They know what to do. We just have to give them the signals and kind of coax them along. So one way we do this in lab is with bioreactors. And that's basically just a fancy home for cells to live in. And so I can take those beating cells that you just saw and mix them together with supporting cells like fibroblasts, and then just inject them into this custom bioreactor. And it'll shrink over time and form a piece of cardiac tissue around these 2 flexible pillars. And these 2 flexible pillars are really important for our design, because whenever you're making an organ on a dish, you want to mimic what's going on in the body. And so for your heart to pump blood, it has to pump more blood and change the force with which it pumps this blood if you're doing something like running or if you're sleeping. And so we need to mimic that by imposing the passive tension of these pillars and making our little heart tissues in the lab also have to work against the force. And then we need pacemaker cells, right? Because there's something in your heart, your pacemaker cells, that tell your heart when to beat and how fast to beat. So we can just add carbon electrodes into our dish and tell the cells when to beat by giving them this electrical signal. So we can then tell the cells how hard to beat, how fast to beat, and when to beat, so we can make these mini training regimens. And just like you would work out any muscle in your body, we make our heart tissues work out, and so every day they're becoming stronger and more mature until they ultimately become this really mature piece of heart tissue that's capable of acting just like your heart does in your body. So then you can use these heart tissues and actually screen drugs. So the ultimate gold standard is, let's see if this actually works on a drug that went to market, that made it through clinical trials and failed. So cisapride's an example of this drug. This drug went to market, was massively prescribed, and then it was found out, unfortunately too late, that there were many conditions where this drug shouldn't have been prescribed, where there were lethal consequences. So unfortunately, cisapride, caused many fatalities and is no longer on market. So let's use our tissue in the dish and expose it to similar, put it through drug screening process. So if we take cisapride at a bunch of different concentrations and we put it on our little tissue in a dish, we can get this EC50 value. So this is the safe concentration where the drug is still working, but you're not inducing negative side effects. We can take that concentration and then add it to our tissues in a dish. So if you add it to a healthy tissue, nothing happens. This drug would be safe. So what went wrong? Well, we're not all healthy populations. We wouldn't really need medicine if we were all perfectly healthy, would we? And so we need to mimic that when we're doing drug screening, mimic our diversity, mimic if we have underlying diseases. And by using organ-on-a-chips, you can do that, and you can make a bunch of little mini us, all of these little mini people in a dish, make our organs, and then screen drugs on them. And so then you start to find out that if you had an underlying disease, you shouldn't take this drug, because at that same concentration that's safe for healthy people, it's actually lethal in you and would induce these episodes of arrhythmia and sudden cardiac death. Or if you're one of the many people who've had a heart attack and have heart disease, you also should have never been prescribed this drug. So if we can gain all this information from these human models, we can then put that into the drug development pipeline and make better, more informed decisions about what drugs should we develop, and also who should we give them to? Who's that intended market? Who will it work for, and who should stay away from it? And so we go back to the whole, pharma needs to make money, right? They're not just going to develop drugs for us because we need them and we want what's most effective. And so by getting that drug to market where it's actually going to work and you're not getting these failures, you're saving pharma tons of money. Anywhere from $100 million to $3 billion for each drug. So there's a saying in the field that says, fail fast and fail quick. Well, with animal models, that doesn't enable that to happen. So using human models, you don't waste time developing drugs that are never going to make it in the long run, because while they weren't toxic in mice, they are in humans. And so if you think of drugs as kind of a common good or commodity, you're not wrong, but they're also products. And they're products, sold and developed by for-profit companies, and these companies have shareholders that they have to answer to. So it's not surprising that there's a lot of drugs that just aren't being developed. You saw the numbers and timelines associated with drug development. Why would you invest in a drug that's not necessarily your sure thing, or doesn't have a group of people who are going to pay the money needed to make up for all those losses you incurred? So unfortunately, that leaves a lot of us out of this drug development paradigm. If you don't have the means to pay, or you're affected by a disease that doesn't affect that many people, you're left out. And this is where organ chips can really help and develop drugs for those who need it the most. For example, for rare diseases, while there are over 7,000 of them, for each individual disease, you might think that it's too rare to invest in. But when you aggregate them all, 10% of us has a rare disease. So could you imagine, one of your friends or you go into the doctor and you finally figure out what's wrong with you, and then you learn that there's no treatment available and no treatment being developed. And 95% of these rare diseases have not one clinical drug or FDA-approved drug in the market. Nothing is being developed for them. Or say it's your child, and 35% of all deaths are due to these rare diseases in infants. Could you imagine how devastating it would be for the doctor to tell you that there's nothing they can do? Your child, the most important thing in your life, your world, that's not important enough to develop a drug for? That's devastating, and we need to change that. So in order to change that, to be that change, we can use these human models and make organs in a dish. So take Timothy syndrome, which is a disease that only ever affects 20 people in the world at a time because it's very lethal, and that's the only number of people you have alive. So we took cells from somebody with Timothy syndrome, and we made their disease in a dish. And then we can mimic the conditions where somebody with Timothy syndrome would experience an episode of pro-arrhythmic episodes or occurrences, and that unfortunately leads to death. But if you have this organ in a dish with the disease, you can start testing a lot more drugs than one of these 20 people in the world. So we started testing drugs, and we found that there are hits. There are drugs that would work for this person. And that's really important to be able to not only model healthy tissue, but these diseased tissues in the dish, so we can start finding cures for everyone, regardless of who they are or if they can pay. Because drug development shouldn't be for the select few. It should be for all. So we can fast track this drug that could work for Timothy syndrome and start getting it, through the clinical trials and into the hands of those who need it the most. And if that's not enough incentive, if just helping people, more isn't enough incentive to develop these drugs, by taking a drug to market that would ultimately succeed, instead of dropping it early on because in a mouse model or a rat model there was a potential risk or a red flag, you can kind of sidestep, see how it works in these human tissue models, and then make a more informed decision. Does it actually have toxic side effects in humans, or is it a species mismatch because I'm using it in a rat, and a rat's not necessarily mimicking our hearts in humans? And so if that's the case and you continue to develop this drug, that's $4 billion in revenue that you're generating. So whatever incentive works for you, the bottom line is that we need to be developing drugs for all, regardless of what affects you or whether you can pay. And so by making these organ-on-a-chip models that are much more predictive and decreasing all the lengthy timelines and high costs associated with drug development, we really start to move towards this paradigm where we can develop drugs for everyone equally, because we all deserve to have drugs, for us, that work for us and that are safe for us. So by using, our cells, our tissues, our organs in a dish, this becomes a real possibility. And so it's my hope that in the future, the next time I go to the doctor with Uncle Billy, we're taking his cells, and then we're making his organs in a dish so we can screen drugs on, Uncle Billy's cancer and find out what's going to treat that. And so when we're developing, his treatment plan and deciding what will work, the doctor already knows. He's got scientific evidence to say, hey, this is the drug that works the best for your cancer. So that's the one we're going to try. And none of this, medicine's not one size fits all. The drug that works best for you or for your friend or for your uncle is not going to work the best for my uncle. And I want to know from the beginning what drug will work best for him. He deserves that. And so let's start to make that a reality. And instead of putting him through all these clinical trials, let's come and give him his cure, because we use these organ-on-a-chip systems to figure it out. And so if we already have the technology available to increase the efficiency with drug development, increase the efficacy, and then increase the equality involved in it, what are we waiting for? Let's start to make this a reality.